Now today in this session we are going to discuss about this gametogenesis. This is in the reproductive system. Gametogenesis. Now what is actually gametogenesis means? Gametogenesis means the formation of gametes. Formation of gametes is called as gametogenesis. Formation of gametes. This is called as gametogenesis. Now gametes are formed in the primary sex organs, testis in males, ovary in females. Now they are gametogenesis takes place in primary sex organs. Now this primary sex organs, testis in males, this is in males, ovaries, this is in females. Now that is in males it is called as spermatogenesis, whereas in females it is called as oogenesis. Now in testis it takes place in males, it is called as spermatogenesis, whereas in females it is called as oogenesis. This is gametogenesis, formation of gametes in both male and females. Now it takes place in the primary sex organs. In males it is in testis. This process is called as spermatogenesis, whereas in females it takes place in ovaries. This process is called as oogenesis. Now first we will discuss here the spermatogenesis which takes place in the testis. Spermatogenesis. Here is spermatogenesis. Formation of spermatozoa in the testis. Formation of spermatozoa in testis. Now in testis, the part where the sperms are formed are seminiferous tubules. This is what we have already discussed, the seminiferous tubules. Seminiferous tubules are the sites for the production of sperms. In the testis, this is the exact position or exact place where the sperms are produced. They are seminiferous tubules. Now seminiferous tubules are present in the testicular lobule. 1, 2, 3 seminiferous tubules present in the testicular lobule, which was cut by Tudica albuginea. That what we have studied. Now in these seminiferous tubules, what are present? Now they are spermatogonial mother cells. Now what are there? Spermatogonial mother cells, or simply spermatogonia, or other simply spermatogonia. Now these are the ones which are present in the seminiferous tubules. Seminiferous tubules are lined by germinal epithelium, which is having cuboidal epithelium. This cuboidal epithelium consists of the spermatogonial mother cells or spermatogonia. Now from these cells the sperms are formed. Now the sperms are formed by the mechanism how they are formed. This is what we are going to discuss now. The spermatogenesis, the process of spermatogenesis, which results in the formation of spermatozoa or sperms. Now it involves the following. Now this process of spermatogenesis takes place only after this is puberty. It means the male child has to reach puberty to take place this spermatogenesis under the influence of secondary sexual hormones, testosterone production by the Leydig cells. Now it means spermatogenesis starts on the onset of puberty. This is 13 to 15 is the age in males where he reaches puberty, that is man reaches puberty, the boy reaches puberty. Now, the spermatogonial mother cells. Now, how this process takes place? Now, this spermatogenesis involves following 3 stages. What are those 3 stages? The spermatogenesis. What are the 3 stages involved in the production of spermatozoa? Here is first multiplication phase, growth phase, then maturation phase. Now, these are the 3 steps or phases through which the spermatozoa are formed. Multiplication phase. Now, multiplication phase, in this phase, the spermatogonial mother cells, they undergo division. That division is mitosis and produces the spermatogonia. Now, here is, for suppose, this is the spermatogonial mother cell. This is the spermatogonial mother cell, or this is spermatogonial mother cell, which is present lined in the seminiferous tubule. Now, this spermatogonial mother cell, by division, it produces 2 types of cells. By division. The division is mitosis, because this is a diploid one, the spermatogonial mother cell. It produces 2 types of cells. What are those 2 types of cells? These are, here is spermatogonia A type, spermatogonia A type, spermatogonia B type. Now, this is 2 types of cells which are produced. Now, this is spermatogonia A type of cells. This is spermatogonia B type. Now, the spermatogonia B type, they divide and redivide to produce large number of spermatogonia of B type. Now, these all are spermatogonia of B type. This spermatogonia B type of cells, they undergo division. This is mitotic division to produce large number of cells. What are these cells are called as? These are called as spermatogonia B type of cells. These are B type of cells. What about this A type? A type act as stem cells or reserve cells. They act as stem cells or reserve cells they act as. Now, whenever required, the A type will produce B type. A type, they undergo division to produce B type whenever required. Now, B type, they undergo mitotic division to produce large number of P type of spermatogonia. Now, this stage is what we are saying multiplication phase. This phase is multiplication phase. Okay. Now, this multiplication phase results in large number of spermatogonia. What are these ones? Spermatogonia. Now, these spermatogonia undergoes, this is growth, another phase here, this is growth. Now, it means these cells become larger. This is growth, they undergo growth. Now, these are called as primary spermatocytes. These are called as primary spermatocytes. Now, these primary spermatocytes, they undergo meiosis 1. Now, this is also 2n, these are also 2n, means diploid. Now, this meiosis 1, after meiosis 1, the primary spermatocytes give rise to secondary spermatocytes. These are two. Meiosis 1 results in the formation of two cells here, two equal cells. Now these are called as secondary spermatocytes. Now these secondary spermatocytes undergo meiosis 2. Meiosis 2 is like that of mitosis. Now they undergo here meiosis 2. Now which results in the formation of 4 haploid cells. Now what are these cells are called as? They are called as spermatids. They are called as spermatids. Now these are the spermatids, 4 spermatids. Now these spermatids, they undergo differentiation. This is morphological differentiation. Structurally they are round, actually they become elongated. Now this is elongated structures. What are these called as? These are called as spermatozoa. These are the spermatozoa. Now these are the spermatozoa. Spermatids have undergone what? This is a differentiation. Now this process is called as maturation phase. Maturation phase. Multiplication, growth and maturation results in the formation of spermatids. These spermatids are changed into spermatozoa by morphological change. Now this spermatids forming into spermatozoa, this is called as Spermiogenesis. Or otherwise also called as spermatoleiosis. Spermiogenesis or spermatoleiosis. Okay. Once again I repeat here, there are spermatogonial mother cells which are lined in the seminiferous tubule. Now these spermatogonial mother cells, they divide by mitosis, produce two types of cells, type A, type B. Spermatogonia type A, spermatogonia type B. Now type A cells are reserve cells or stem cells. Whenever required, they produce type B cells. Then these spermatogonia B type cells, they undergo mitosis and produces large number of spermatogonia of B type. Now these spermatogonia which are formed, this process up to here, this is called as multiplication. Now the spermatogonia show increase in the size. Increase in the size is what we are saying, increase in size. This is called as growth phase. This is the growth phase and it becomes primary spermatocyte. Now up to here, these are diploid. Spermatogonia, spermatogonial mother cells as well as primary spermatocytes, they are all diploid. Now after growth phase, these primary spermatocytes, they undergo meiosis 1 to form secondary spermatocytes. They form here secondary spermatocytes by undergoing meiosis. Now these secondary spermatocytes, which are now haploid. Now each secondary spermatocyte undergo meiosis 2. Meiosis consists of meiosis 1, meiosis 2. Now they undergo meiosis 2 to form 4 equal sized structures, what they are called as spermatids. These are also N. This is haploid. These are also haploid. Now these spermatids, which are round in shape, they get elongated. Now this morphological change is what we are saying spermiogenesis or otherwise spermatoleiosis. Otherwise they are also called as spermatoleiosis. Spermatoleiosis. Now the spermatids change it into spermatozoa. Now these spermatozoa embedded their heads, the sperm heads embedded in the serotoli cells to get nourishment and maturation further. to get the swimming ability. The spermatozoa, their heads embedded in the Sertoli cells for getting further nourishment and maturation, and they become discharged into the lumen from the head. This is Sertoli. From the Sertoli cells, they are discharged into the lumen. Now then they are called as sperms. Okay, this process is called as dash. Now this process is called as spermiation. What we are saying? Spermiation. What is spermiation? Spermatozoa after attachment to Sertoli cells get detached into lumen. Now this process is called as spermiation. Okay, now this is what happens in the seminiferous tubules. In the seminiferous tubules, multiplication, growth, maturation. Then these spermatozoa, their heads get attached to the Sertoli cells. And then they get nourishment, get maturity, then they detach from their head from the Sertoli cells, coming into the lumen of the seminiferous tubule. This is called as spermiation. Release of sperms from the Sertoli cells is called as spermiation. Release of sperms from the Sertoli heads, from the Sertoli cells, is called as spermiation. Now this spermiation process takes place later. Okay, now this is how the sperms are produced in the seminiferous tubule. Now diagrammatically we will show here, diagrammatic representation how this process takes place. Here is, this is the seminiferous tubule. This is, now we are showing here, this is a cut end here, a cut end of the seminiferous tubule. Now this is the seminiferous tubule, a cut end. Now here, these are the cells, spermatogonial mother cells. Actually this is cuboidal epithelium. These are the spermatogonial mother cells, which are lined in the seminiferous tubule. Now in the previous class also what we have said is, these cells, spermatogonial mother cells or oogonial mother cells, they are extragonadal. Extragonadal means from the yolk sac at the 5th month they migrate from the yolk sac into the testis, whereas in ovaries, extragonadal in origin. Their origin is extragonadal, means they are not formed in the gonads, not in the testis, they are extragonadal. Now this is a spermatogonial mother cells. Now the spermatogonial mother cells undergo division to produce spermatogonia. Now here these are the spermatogonia. Now these are the spermatogonia. Now these spermatogonia, they differentiates into primary spermatocytes. Now here is the primary spermatocyte. Each one gives rise to 2 primary spermatocytes. Now for suppose this one has given rise to primary spermatocytes. Okay. Now this is primary spermatocytes diploid. Now each primary spermatocytes undergo division to form, these are the secondary spermatocytes. These are the secondary spermatocytes. These are the secondary spermatocytes. Now this is, here we are showing this is spermatogonial mother cells. These are the spermatogonial mother cells. Then these are the spermatogonia. Now these are the primary spermatocytes. Now these are the secondary spermatocytes. Now this primary secondary spermatocytes, they undergo here, this is meiosis 2 to form, this is the spermatids. What are these structures? Spermatids. Now these spermatids, they get elongated here, this is they get elongated, this is how they get elongated. Now then they are called as spermatozoa. Now their heads, now this is the head, their heads get attached to the serotoli cell. Now I am showing here one serotoli cell, this is the serotoli cell, a tall cell, what we call it as serotoli cell. Now this is the heads of the sperms or spermatozoa, they get attached to the serotoli cells. For what purpose they are attached to the serotoli cells? To get nourishment, to formation of acrosome, head region, they get the ability to swim, everything. They get, this is nourishment from the serotoli cells. And then from this serotoli cell, these spermatozoa, they get detached, their heads get detached from the serotoli cells and they enter into the lumen. Now this is into the lumen. Now then they are called as sperms. Okay, this is what we have shown there. This is spermatogonial mother cells, this is spermatogonia, these are the primary spermatocytes, these are the secondary spermatocytes. Now which undergo division to form spermatids. Spermatids differentiates into spermatozoa, their heads get attached to the serotoli cell, this is the serotoli cell, and derive nourishment, they undergo maturation and finally they get detached from the serotoli cell, this is called as spermiation, what we call it as detachment of the heads of the spermatozoa and open, this is released into the lumen, this is called as spermiation. Okay. This is how the spermatozoa or sperms are released into the lumen. Now how the hormonal control, how this hormonal control of this spermatogenesis takes place, hormonal control. Here is the hormonal control of spermatogenesis. Now for next here, this is we are showing the hormonal control of this spermatogenesis. This is hypothalamus, this is the hypothalamus, this is the pituitary, anterior pituitary, this is posterior. Now this is anterior pituitary gland. Now this is hypothalamus. Now this is anterior pituitary. Now hypothalamus releases a hormone called as GnRH, gonadotropin releasing hormone. Now this released into the anterior pituitary. Then anterior pituitary will release two hormones, that is FSH, LH, follicle stimulating hormone, luteinizing hormone. These are the two hormones which are released from the anterior pituitary under the influence of GnRH. Now follicle stimulating hormone and luteinizing hormone. FSH, follicle stimulating hormone, it acts on which cells it acts on? It acts on the germinal epithelium. Whereas luteinizing hormone, it acts on, luteinizing hormone otherwise also called as ISCH, that is interstitial cell stimulating hormone, it is also called as ICSH, interstitial cell stimulating hormone, ICSH, it is also called as LH, also called as. Okay. Now FSH, it acts on This is a seminiferous tubule. This is acting on seminiferous tubules. Whereas LH, it acts on Leydig cells. Leydig cells are otherwise also called as interstitial cells of Leydig, or interstitial cells, they are also called as. Now seminiferous tubules means here FSH is acting on the spermatogonium for the production of spermatogenesis. FSH is acting on the seminiferous tubule, means seminiferous tubule consists of two types of cells. What are those two types of cells? One is germinal epithelium, which consists of spermatogonia. One type of cells are spermatogonia. Now other type of cells are ceratoli. Now these are the two types of cells on which the FSH act. FSH act on seminiferous tubule, which consists of spermatogonia, ceratoli cells. Whereas LH or ICSH, it is also called as, it acts on Leydig cells or interstitial cells. Now Leydig cells, under the influence of LH, it will produce androgens. Androgens are nothing but here testosterone. This is the male hormone. Okay. Now spermatogonial mother cells, they undergo division. Now this is what happens. This is called as spermatogenesis takes place. Spermatogonia undergoes spermatogenesis to form spermatozoa. Whereas it is also acting on ceratoli cells. Then ceratoli cells will release, these are the hormones. It releases ABP, androgen binding protein. Then another one, inhibin, and another one, anti-Mullerian hormone. Androgen binding protein, inhibin, anti-Mullerian hormone. Now these are produced by ceratoli cells. The spermatogonia undergoes spermatogenesis to form spermatozoa under the influence of FSH. Now ceratoli cells produces ABP, inhibin, anti-Mullerian hormone. Okay. Now here is, for suppose this is the seminiferous tubule. Now it consists of, these are the cells. What are these? Ceratoli cells. Now what are these cells? These cells is spermatogonial cells, which produces sperms. This is the germinal epithelium, and here and there are tall cells called as ceratoli cells. Okay. Now it releases ABP, androgen binding protein, and makes this testosterone. Androgen means this is male androgen is testosterone. Testosterone is taken in by the ABP, which is produced by ceratoli cells. Ceratoli cells producing ABP, androgen binding protein, binds with androgen for the process called as spermatogenesis. Then inhibin, if there is excess, now this is excess of spermatozoa in the seminiferous tubule gives a negative feedback. This is inhibin is produced by the ceratoli cells. Ceratoli cells produce inhibin when there is excess of spermatozoa in the seminiferous tubule, and it gives message to the anterior pituitary. This is a negative feedback mechanism. Listen what I am saying. First, this is a ceratoli cell is producing ABP, androgen binding protein. Now this testosterone is an androgen. This ABP attract the testosterone into the seminiferous tubule for the process called as spermatogenesis. 1. When the sperms are more in the lumen, it produces inhibin. This inhibin sends a negative feedback mechanism to the If this is anterior pituitary not to release these hormones, so that no excess sperms are produced. Then if this excess of testosterone is produced, now this testosterone is more inside. Now this testosterone which is produced, it also provides here negative feedback to the anterior pituitary, another negative feedback. So this is FSH and LH, both are not released. Simply inhibin is produced only when the excess of sperms are present in the seminiferous tubule. Then it sends a message, inhibin will send message. This is a negative feedback mechanism not to produce this LH. When testosterone levels are high, then it will send again a negative feedback to the FSH not to be produced. Okay. Then anterior pituitary, the GnRH is not released, maintains the spermatogenesis. Now this is how hormonal feedback mechanism. Simply what we are saying, this is hypothalamus which is releasing GnRH, gonadotropin releasing hormone, into the anterior pituitary. The anterior pituitary will release two hormones. What are those? Gonadotropins. These two are together called as dash gonadotropins, FSH, LH. LH also called as ICSH, interstitial cell stimulating hormone, because it is stimulating interstitial cells. Now FSH acts on seminiferous tubule for the process spermatogenesis, and also acts on Sertoli cells. Then Sertoli cells releases three types of hormones depending upon its usage. ABP will be released, attracting the testosterone into the seminiferous tubule for spermatogenesis. If sperms are more, inhibin is produced and it gives negative feedback. If testosterone is more, it gives again negative feedback. Now Mullerian, this is an anti-Mullerian hormone. It suppresses the Mullerian duct. Mullerian duct is nothing but seminal vesicle, that is uterine tube in the females. This is anti-Mullerian, suppresses the Mullerian duct, nothing but a seminal vesicle. Okay, this is how, actually this is Mullerian hormone suppresses the female reproductive activity. Female reproductive activity in the males. Okay, now this is a hormonal control of spermatogenesis. Is it clear? This is a GnRH releasing gonadotropin FSH, LH and ICH. Okay, this is here terminology what we use. The terminology what we use. Spermiation. Now spermiation means release of sperms from the Sertoli head. Release of sperm from Sertoli head. Sertoli cells. That is, head of the sperm is released from the Sertoli cells. Then another word. This is it may not be visible here. What is this one? Is spermiation. Spermiation means release of sperm from Sertoli cells. Then semination. Now semination means release of sperms from the testis. Release of sperm from testis. This is a semination. Then emission. This is release of sperms from ampulla. Release of sperms from ampulla. Then ejaculation, release of sperms or discharge of sperms to the outside, discharge of sperms outside the male body, sperms outside the male body, then insemination, this is discharge of sperms into the vagina of the female. This is discharge of sperms into female vagina. Now what we are saying, spermiation, release of sperms from the Sertoli cells. Now this is the Sertoli cells, these are the sperm heads embedded. Now this release of this sperm, this is spermiation. Semination. Now semination means release of sperms from the testis. Now this is the testis. Now these are the testis. Now these are the epidermis. Now this release of sperms here into the epidermis, that is out of the testis. This is semination, release of sperms from the testis. Then emission, release of sperms from ampulla. Now there is vas deferens form here, there is ampulla. Now there will be seminal vesicle attached here to form ejaculatory duct. Now this is ampulla. Now the sperms which are released from the ampulla is called emission. Ejaculation, discharge of sperms outside the male body through penis. Through penis sperms are released out. This is ejaculation. Insemination, discharge of sperms into female vagina. It is into the female reproductive tract. This is insemination. This is also the order. First spermiation, then semination, then emission, then ejaculation, then insemination. Arrange the following in their order of release of sperms. First comes is spermiation, then comes semination, then emission, then ejaculation, then insemination. Okay, this is how the sperms are released from various parts of the male body. The different names are given here. Now with this, we will complete this session. In the next session, we will discuss semen as well as structure of spermatozoa. Thank you.